In this lecture, we're going to start our journey into the nervous system as a whole. So we've covered the cells that make up the nervous system and how they function from an electrical and chemical basis. And now we're going to start working our way inward into the nervous system, and we'll continue working up for the rest of this class. So we're going to start with peripheral nerves here, those things that are connecting the central nervous system to the rest of the body. And so there's a lot that we've already covered. You know, we're still made up largely of axons. That's what we're looking at here. This big N here, that's the node of Ranvier. So here's where we'll have our ion channels and recharge our action potential. There's a couple of myelinated internodes here so that we quickly conduct information along these axons. And peripheral nerves are largely made up of axons. Of course, there's a few other things in there, and we'll touch on those as we go over the structure of the nerves in the first part. And then the orderly arrangement with which the nerves come on or come off of the spinal cord, however you want to think of it, that's what's going to create these different spinal levels that give us our dermatomes and myotomes. So the peripheral nerves are there really to connect the central nervous system, and for the most part, we're thinking about the spinal cord right now, with the rest of the body. Now, that said, cranial nerves are also in this group here, but they'll get their own lecture. Okay, a couple of terms here, afferents and efferents. Afferents arrive into the central nervous system, so they're sensory in nature. So when you think afferent, just think arrives. And that's what makes them sensory. Efferents exit the central nervous system, so they're motor in nature. And there's a couple of types. There's those lower motor neurons in the ventral horn. Those are going to head on out the ventral root there, and they'll innervate some muscle. There's also autonomic neurons in more intermediate areas, the lateral horns that we're not seeing here, but we will in a little bit. Both of those go out of ventral nerve roots. The ventral root then combines with the dorsal root at the intervertebral foramen, and they form some kind of a peripheral nerve. The sensory aspect is coming in the dorsal root. Those neurons live in the dorsal root ganglion. They're going to come in. They might synapse with some interneurons. Here we're getting some kind of a reflex, and they might just project on up. They'll get their own time later. But you'll notice where these two things are converging is right there, and that's at the intervertebral foramen, or the hole, a foramina, between the vertebrae. Just that simple. So ventral, dorsal, coming together at the intervertebral foramen. Now, because this is a site that passes between bone, this is obviously a potential site of injury that we'll see in a little bit, and we'll hit that at the end of the lecture. When we compress the nerves, we're going to get sensory and motor deficits as a result. Okay, once we exit as a spinal nerve, we start branching. And the first branch here is this meningeal branch. So, okay, again, we're our dorsal root here, our ventral root. We've come together at the intervertebral foramen, and now our first branch point is the meningeal branch. Notice what we're hitting. The anterior aspect of the dura, not posterior. The posterior dura remains uninnervated. That way we can do our spinal taps, right? We don't have to worry about numbing up the dura or anything like that. The anterior aspect is what's innervated by the meningeal branch. And this is the thing that gives us back pain. So it's getting the dura, it's getting the posterior longitudinal ligaments, it's getting the intervertebral discs. Notice it's just getting the anterior aspect of things. And so if we have any kind of bulging disc or any irritation, this will give us our back pain. Okay, so there's one possible branch. Then we continue along, and we've got another branch point. Okay, back to this illustration. Here we have our sensory and motor fibers coming together, and then they branch to form either the, let's make it black, dorsal or ventral rami. Okay, so for dorsal, this branch here, notice it's mixed. It's got sensory, the blue, and motor, the red. This is going to head on back and hit the muscles and skin of the trunk and the posterior aspect of the trunk. So this will help maintain our posture. It'll let our backs sweat, let us feel what's on our back. The ventral ramus is much larger because not only is it serving the trunk, in this case the anterior segment, it's also hitting the limbs. And boy, the limbs account for a lot more of our body collectively than just the back of the trunk. Okay, now along the way, in order for us to innervate the limbs, we're going to form a number of plexuses, which you are certainly well aware of. Here they are again for your pleasure. One that we really want to focus on is the brachial plexus because this innervates the upper limb. These things are handy, and you will definitely want to know your brachial plexus. We'll touch on that later. There are a number of other plexuses. We got a lumbar and sacral plexus down there for the lower limbs. These are nice. Don't sleep on the cervical and coccygeal plexuses either. These are important. But if I had to pick one to know, it would be the brachial plexus, and we will spend time on that in later lectures. But what's going on here, all those ventral roots are converging and diverging to form the specific peripheral nerves of the limbs. And that's why these peripheral nerves correspond to more than one spinal level because of all the mixing that happens at the plexuses. An optional branch for our peripheral nerves are the communicating rami. So it's still some kind of a branch here, just like the dorsal and ventral ramus. Before we form those, we may very well have a communicating ramus to connect us with the paravertebral ganglia. We're only going to find these in the thoracic, so T1 to L2 segments of the spine. That's where those preganglionic sympathetic neurons living in the lateral horn, that's what this is here. Notice that additional horn. So we still have the ventral horn, the dorsal horn, and now the lateral horn contains these preganglionic sympathetic neurons. These are going to head on out the ventral root just like they did earlier. We're going to converge at the intervertebral foramen, and then, don't forget, we have a meningeal branch, but we don't care about that right now. We head in the white ramus, and it's called that because we are myelinated right now. Myelin, being mostly fat, appears white. Okay. Then we have another loop coming back, the gray ramus. And it's gray because these fibers are not myelinated. Okay, so there's one option. We come on in the white ramus, we synapse, and then this postganglionic neuron here, this red one, heads back into the spinal nerve via the gray ramus. And that's going to follow along the dorsal, the ventral rami, and that'll innervate the sweat glands, erector muscles, and blood vessels of the limbs and the trunk. Option one. Okay, another option. We could form a sympathetic nerve. Okay, so we come in and we're going to synapse, but maybe not at this level. Maybe we head on up to one of those cervical ganglia, right, or one of those lower lumbar ganglia or something like that. And here, because we don't have a gray ramus as an option, we're going to head on out and form ourselves a sympathetic nerve. For example, ciliary nerve to innervate the iris, or the cardiac nerve to innervate the heart. So there's another option. It's very similar in nature, but we're just simply not going back to some, mixed somatic, motor, and sensory fiber. The third option is we head on in the white ramus, and we don't synapse at all. We just head on out as one of those splanchnic nerves. And what we're going to hit are postganglionic neurons that live somewhere else. Rather than being in the paravertebral ganglia, also called the sympathetic chain ganglia, we're going to go over to the prevertebral or collateral ganglia that are scattered around out there in the body. There's a handful of these that we'll cover when we talk about the autonomic nervous system. But it's the same idea. Preganglionic innervates postganglionic. They're just in a different location. Therefore, the splanchnic nerves are still myelinated. Okay, so there's an option. Either way, no matter what you choose, you're in some kind of a spinal nerve, and they kind of look the same in a cross-section, right? If we cut through a nerve here, we see that we got a whole bunch of axons and there's a bunch of connective tissue holding this whole thing together. And that connective tissue is collectively called the mesoneurium. So that's all this is. Mesoneurium is just connective tissue. Think of this like your meninges. And this is indeed continuous with at least some of the meninges. The epineurium is continuous with the dura mater, for example. And the perineurium is somewhat continuous with the arachnoid. Endoneurium, I think, is its own thing, but what do I know? Okay, the epineurium is on the outside. It's around the nerve. Hey, the name makes sense. Just like the dura, it's made of the same stuff. It has the same function. This is physical protection. So everything around and within the nerve here is epineurium until we get to these bundles of axons called fascicles. So here's a big old fascicle. Here's another one, another one. These are blood vessels. Okay, the perineurium, highlighted here, is what surrounds these fascicles. Much like the arachnoid, this is a diffusion barrier. So these perineurial cells have these tight junctions between them so no diffusion can happen. Anything nasty in this blood vessel can't diffuse into the fascicle. This creates the nerve tissue barrier. It's a barrier between all these nerve fibers in here and the surrounding tissue. There's also a blood-nerve barrier created within the fascicles. Also within the fascicles, we have endoneurium. Think of this as just kind of some jello. It's there to give the axons and blood vessels something to rest in. And that's it. So we got our 3 layers, epineurium, outside, that's like the dura, perineurium, that's around the fascicles, kind of like the arachnoid, it's a diffusion barrier, and then the endoneurium is within the fascicles, allowing the axons and blood vessels to have something to float in. Now you'll also notice in cross-sections of peripheral nerves that we have a variety of axon diameters. Some axons are really big, and I've put those at the top. You'll notice this is arranged from largest to smallest. And the biggest ones are the A fibers, and then they get a fancy little Greek letter. How nice is that? What you should notice here is that the largest fibers have something to do with movement. It could be that they are lower motor neurons, innervating extrafusal muscle fibers and letting us move. Or maybe they have a sensory function in the tendons or muscle spindles, creating nice reflexes for us. But everything that has to do with moving our muscles, actually generating movement, that is the fastest. We want to get our body in the right place, now, not later. And why is this the fastest? Because they are myelinated, like pretty much all the others, and they have the largest diameter. By decreasing that internal resistance, it facilitates current flow within the axon. Just a little bit smaller, and thus just a little bit slower, we have our A beta fibers, and these are sensory in nature. Notice we're still getting to the spindles, right? Muscle spindles are still here, but these are more important with knowing where our body is, right? Proprioception. And then we have all the encapsulated nerve endings in the skin, right? Joints and muscles, so we can tell where our body is. So we have tactile sensation below that. The A gamma fibers, these are from gamma lower motor neurons, which are a little smaller and thus a little bit slower. They control the intrafusal fibers and set the desired length of the muscle. Then we get into the A delta and C fibers. I'm going to skip over B for now. These are the pain and temperature sensing fibers. So they are the bare nerve endings out there in the skin, muscles, and joints. They give us pain. A delta, a little bit larger and myelinated, and so quite a bit faster than C fibers. A delta will give us more sharp pain. C fiber will give us the more dull, burning type pain. Now, I glossed over B. B fibers are the preganglionic autonomic neurons, so they target postganglionic neurons, which are C fibers. So there's A delta and C for pain and temp, and there's B and C for autonomic function. The postganglionic neurons, of course, target viscera, glands, blood vessels, more on that when we cover the autonomic nervous system. The big takeaway here is that if you are larger and myelinated, you conduct information much faster. What do we conduct the fastest? Motor output. Then we have sensory. For sensory, we have tactile sensation much faster than pain. This makes pain a bit slower to get there, but it also lasts for a bit longer. I think the logic there is that you have to feel the pain for longer, and you thus hopefully learn from it and don't do that thing again that hurt you. And then down here at the very bottom, autonomic function. This is quite slow and long-lived, just like pain. So we get these long-lived changes in, blood pressure and how much we sweat and things like that. Now, we've talked about myelination already, and this is a wonderful thing. But the thing is, most Schwann cells don't actually myelinate axons. They're what we call non-myelinating or REMAC Schwann cells. Everything needs two names. You can see this is the majority of Schwann cells. I'm not sure if it's 80% spot on, but, close to that. These still surround axons. So here we have a REMAC Schwann cell, a non-myelinating Schwann cell. Here's its nucleus, and it's surrounding multiple axons, but it's not actually myelinating them, right? It's not wrapping it in layer upon layer of myelin like we see over here. It's not getting rid of the ion channels in the axon. It's just getting a little bit of physical isolation. So the action potentials in this axon don't, excite its neighboring axons. So there's a little bit of physical isolation, but there's also quite a bit of work going on too. So what do non-myelinating Schwann cells do? Just think of them as astrocytes in the periphery, right? They're still there to soak up that potassium, making sure that this action potential that's going on in this axon doesn't influence any action potentials over here in this axon. And once we fire an action potential, that ain't free. We got to provide some metabolic support. And just like astrocytes, these are connected by gap junctions. So this non-myelinating Schwann cell is connected to another one, and they work together. All right, let's make sure we got something out of that. So here we can see a cross-section of the spinal cord here. Hopefully we're picking out the dorsal horns back here, the ventral horns in the front. That big old vertebral body is kind of a dead giveaway. Dorsal root ganglion over here is a giveaway. Dorsal root, ventral root. These come together at the intervertebral foramen. That's this big old hole here. And then we branch. We're omitting the meningeal branch here, but we're forming the dorsal ramus. This will go and hit the back. That makes sense. Dorsal goes to the back. And the ventral ramus here is going to head on out and get the front and the limbs. Okay, what we should keep in mind is that once we mix our dorsal and ventral roots, we are now a combination of sensory and motor. So we have mixed sensory motor in the dorsal ramus and the ventral ramus. The dorsal root is purely sensory. The ventral root is purely motor. The ventral horn contains lower motor neurons, and the dorsal horn contains sensory interneurons. So when is it both? We're always mixed when we're in the periphery. Once we exited the spine and we're in some kind of a peripheral nerve, it's mixed. We keep motor and sensory information together we keep motor and sensory information separated in the central nervous system, where they have their own distinct tracts. More on that in later lectures. Okay, and then figure 2 down here, we've got a cross-section of peripheral nerve. The epineurium is all on the outside. It's kind of falling apart here. That happens sometimes. This is probably just an artifact from fixation. But there's epineurium, there's epineurium, there's epineurium. Okay, so where do we have perineurium? Just surrounding these fascicles. Just along the outside of the fascicle is the perineurium. Everything inside is the endoneurium. And of course, the fascicles are these big old circles, these distinct bundles of axons, and some blood vessels too. Alrighty, so we've got our nerves now. They're coming on and leaving in an orderly fashion as we move from high to low in the spinal cord, and that's what gives us our spinal levels. Okay, dermatomes you're familiar with. These are still just strips of skin that correspond to specific spinal roots. Okay, so if we're coming off C5 to T1, somewhere in that ballpark, that's the upper limb. It's great to have some idea of ballparks here, right? My shoulders would be C4, then I go upper limb. I don't go from shoulders to trunk. I go upper limb first, then I'm in the trunk, and notice T. Thoracic goes all the way through the trunk. 4 for the nipples, 10 for the navel, right? This will give you some rough ballpark there. Once we get into the legs, legs, we're in the lumbar spine, but just on the anterior portion. Because notice, we're not going from head to toe. No, we're going from head to tail. So we go down the front of the legs first, just like we went down the, we'll call it front or lateral aspect of the upper limbs and wrapped back up. We're doing the same thing in the lower limbs. So lumbar till we get to the soles of the feet. Hopefully you can figure out a way of remembering that the soles is where we go to the sacral spine and we wrap up the back. Again, head to tail, not head to toe. This body plan emerged not in bipeds like us, but in quadrupeds, most other animals. Now keep in mind, if we say, look, T4 is right here at the nipples. Okay, sure, but there's overlap. T3 is going to get in there a little bit. T5 will get in there a little bit. So just keep that in mind. If we're actually going to have a dermatome disturbed, we need to get that segment and probably the one above and below. We'll need about 3 segments affected before we actually notice anything. Because while this map looks wonderful and clean, there's a lot of overlap in the body. We can see dermatomes pretty nicely, though, with shingles. So sure, we have a lot of overlap, but if you have a shingles outbreak, this is going to show you exactly where all the neurons in that dorsal root are innervating, because that's where the virus lives. So shingles are caused by a type of herpes virus, the varicella-zoster virus. This infects us and then stays dormant in those dorsal root ganglion neurons. So this is going to live there in the dorsal root. And somehow they get reactivated, caused by some kind of stress, whether it's mental or physical, right? So some kind of illness or heck, aging just makes everything worse. The virus reemerges and gets released from the sensory neurons, infecting the skin that is innervated by that sensory neuron and creating these blisters, showing us the dermatome here. We're just below the nipples. Let's call it T5, somewhere in that ballpark. Myotomes are kind of like dermatomes, but it's not skin, it's muscles. Let the name work for you. So this would be the set of muscles that corresponds to some nerve root. And again, we have a nice general rostrocaudal mapping. If you're near the top, you're going to hit structures near the top. So the muscles in the shoulder, that's going to be C5. If we move a little further down the spine, C6, we're moving a little further down that upper limb. We're wrapping back up the medial aspect there, right? Remember that. So same thing with dermatomes. So C7 will hit the elbow there, right? Because that's getting the triceps or something like that. Moving on down, if you're in the lumbar and sacral spine, now we're in the lower limb. So that's kind of nice. The devil's in the details, though, and oh my goodness. What we find, just like with dermatomes, there's a lot of overlap. Great example, let's think about our lower limbs. Okay, I want to flex my hip. I think this is flexion. That would be L2 and 3. Both of those assist with that movement. I want to extend it, L4 and 5. Okay, cool. So some set of muscles will be innervated by more than one root. This should make sense. We still have those plexuses where the ventral rami mix, converge, and diverge to form the peripheral nerves. Okay, what about the knee? I can extend it with L3 and 4, and I can flex it with L5 and S1. Well, this makes sense, right? Remember your dermatomes. Higher up, if we're in the lumbar spine, that'll be more front of the leg. Sacral spine will be more back of the leg. So that kind of makes sense. But what you'll notice here also, let's say I'm L3. What am I doing? I'm flexing the hip and extending the knee. If I'm L4, I'm also extending the knee, but I'm not flexing the hip anymore. Now I'm extending it. So there's a lot of overlap. And while we do follow a nice general rostral caudal mapping, it's a little messy. But you're smart. You'll study it a few times. You'll get it, no problem. Now, as I mentioned earlier, if we have any protrusion into the intervertebral foramen, we can pinch a nerve. We can get a radiculopathy. It might be a herniated disc or these bony outgrowths here. But once we compress that nerve root, this is going to cause pain, of course, right? We might stimulate pain fibers. There's a number of ways to do that. We'll cover it later. We might impair motor function, creating weakness that follows along that myotome. We might have sensory loss that follows along the dermatome, just like the pain. And of course, we're going to weaken our reflexes by weakening the lower motor neuron. We can tell which root is impacted if we know our dermatomes and myotomes. Here's a little table for you. I'm giving you the percent radiculopathy. This would be for cervical radiculopathies on top, lumbosacral radiculopathies on the bottom. That's why this adds up to way more than 100%. Okay, C5, okay, that should be getting some of our shoulder, that upper arm, the lateral aspect. We know our dermatomes. I don't even need to click back at this point. You got it. The reflex, the biceps brachii, I think it's called. Biceps. So we get down a little further, the brachioradialis here, that's more C6. Now granted, C6 contributes here, but mostly C5, vice versa. We get a slightly different sensory pattern though. So knowing both your myotomes and reflexes as well as your dermatomes will help you out here. If we move further down the spine, we're moving further down the arm. So now we get down into the hand, we're getting digits 1 and 2. If it's digit 3, that's got to be a different root, and thus it'll be a different reflex. So take some time, make sure you understand rough ballparks for dermatomes, myotomes, and reflexes at the different spinal levels. And that's kind of what this is going to ask you to do. I'm not going to walk through this in great detail. Just remember, dermatomes are those bands of skin. They correspond to a spinal level. Myotomes are collections of muscles. They correspond to a spinal level. Different spinal levels can contribute to multiple spinal nerves, and we'll see a great example of this later on when we cover this in the lab section. And get a good idea of what are the dermatomes, right? What are the levels for these things? If I'm in the neck, I should know that I'm definitely cervical and I'm kind of high up in it, C2, C3, something like that. C4 will be the shoulders. If I'm in the upper limb, just think brachial plexus, C5 to T1, maybe a little T2, right? If I'm in the trunk, it's going to be thoracic spine, no doubt about it. Nipples, generally T4, navel, T10, something like that. Once I get into the legs, the front of them are lumbar, the back of them are sacral. If I'm in the anterior aspect of the knee, for example, right, I should know that that's somewhere about L4-ish, somewhere in that ballpark. If it's the posterior aspect, it's more sacral, S2, right, something like that. But definitely getting lumbar in the front, sacral in the back, that's a good thing to have. Knowing your reflexes, pretty darned important. Biceps, C5, brachioradialis, C6. Both of these are kind of doing the same thing though, right? We're bending the elbow. Triceps, C7. Patellar tendon, L4 is the big one there. Hey, same thing as the anterior aspect of the knee. That's kind of cool that these things go together. The Achilles tendon, we're down there more, S1, S2, somewhere in that ballpark. But we'll cover that later. If anything on this is confusing, enter it in the questions box and we'll chat about it in class. See you later.